I think hopefully that will become apparent as I talk through some of what we do. But I guess, well, that remains to be seen. Hopefully it becomes obvious. So just a little bit about my story. So I did my PhD in the lab of Nicholas King at the University of Sydney. My whole deal at the time was looking at mouse models of viral encephalitis, and specifically looking at the bone marrow. The hematopoietic system in the bone marrow reacts to something like a viral infection in the brain, and it changes the way it responds depending on the kind of virus or the kind of pathogen that's involved. So I started doing flow cytometry panels, which kind of got bigger and bigger, and then I got to go and do a stint at Stanford for a brief period of time to learn how to use the cytoff around the time that we were getting our first cytoff here in Sydney. So that was quite fun. I got to kind of, go from sort of 15 color or so flow to kind of towards the cytoff. At the end of that, at the end of my PhD, I joined our core facility here as a high dimensional specialist. Essentially what that means now is that I can work with users to try and help them incorporate high dimensional cytometry technologies into their projects, which is quite a feat, but it's also quite a lot of fun. And as part of that, I get to lead a research effort to applying these in a lot of different contexts. The most recent example of this has been doing this with COVID. In this case, this was mainly doing the analysis of the data rather than running the samples, which were run in Melbourne as part of our sort of major wave that happened down there. But something that's become apparent throughout my kind of journey so far is that as our technologies improve and get better and better, we can measure more things, measure more samples. What ends up happening is we end up generating these data sets, which can be really quite difficult to manage. Either because the number of things we're measuring about each cell are becoming just a little bit ridiculous, or the sheer number of cells we're analyzing becomes a bit of a problem. So what I just wanted to briefly introduce is some of the challenges around single cell data and the way we try to analyze it. So we can look at the range of single cell technologies we have available, and these kind of go along this sort of spectrum all the way from sort of conventional flow cytometry with high dimensions. We have spectral, cytoff, kind of all the way through to various forms of single cell RNA sequencing. And these all have different features about the technologies and what they're useful for. And I've tried to summarize some of this on this weird radar plot thing here. Flow cytometry, for the most part, it's still low dimensional in the grand scheme of things. We're only measuring up, nowadays up to around 40 things about each cell, 40, say, antibodies per cell. But it's a cheap, high throughput system, and the ease of entry is quite good. So it's quite easy to get people sort of started with this technology. There's flow cytometers in every lab. There's technicians all over the place, and so that's reasonably straightforward. Cytoff, we're measuring more parameters more easily, but we're running things slower. It's usually, at least in our experience, a more expensive technology, but that does then find a niche in a lot of, say, clinical applications where you do just want to be able to measure as many things as possible at once. And your runtime, it doesn't matter if things are running a little bit slower because you're doing this on patient samples as they arrive. As we move through to things like single cell sequencing, it's a very low throughput technology where we're talking tens of thousands of cells up to hundreds of thousands or maybe some millions in some of the Cell Atlas projects. But that's kind of where that stops in terms of individual experiments, whereas millions of cells in flow cytometry is a routine thing that only takes a couple of minutes. So when we look at the data that's generated from this, fundamentally, we're talking about very similar ideas. We're measuring a number of cells, and on each of those cells, we're measuring a number of features. This might be proteins or transcripts, whatever it is. And we sort of expand these out into dimensions. As we increase the number of features being measured, we sort of increase the complexity of what happens with the data. How many different possible populations or how many different possible cell states could we find? And as we increase the number of cells, we're able to identify very low frequency subsets of cells. We're able to find generate better statistics because we increase the number of samples in our experimental groups, and we can also run more samples together and decrease the number of batches that we might need to use for an experiment. So we sort of create this paradigm where we've got more cells per second versus cost per cell, but this paradigm is changing as these technologies are getting better. Now, what happens with the data is that on this sort of single cell sequencing side, I guess because single cell seq has existed for a while now, and the only way to be able to analyze that data in any meaningful way is using computational methods, this side of things is really quite well developed because there is just no choice but to do this in a particular way. Whereas I think on the sort of, the flow and the side-off side of things, this has been a little bit slower in terms of its uptake because we can, gate flow cytometry or site-off data. And we've been sort of slowly coming around to incorporating this into our work. And there's a range of tools out there which kind of facilitate this sort of work. But one thing that we found was that kind of dealing with the sheer amount of data in terms of number of cells was a real challenge. A lot of tools are very good at providing sort of, really good clustering options, dimensionality reduction options, batch alignment options, but a lot of them really struggle when you put in millions and millions of cells into an individual experiment. So in a couple of the experiments I'll show, we have data sets that are 40, 50, 60, 100 million cells in an individual experiment, and that's where we need to be operating in the kind of flow or site-off space in terms of our analysis. So what we came up with was an R package which was designed explicitly to do high-dimensional single cell analysis, but of large data sets. Data sets where there's high memory pressure, I guess, if I could put it this way. So we called it Spectre. That's not an acronym. We just thought the name sounded good. And so it's a computational toolkit in R so that we can integrate, explore, and analyze our high-dimensional cytometry data. And just very briefly, this is kind of the overall workflow that we talk about with this package. We start with kind of data pre-processing, which is really quite boring content, but it's about getting the data prepped so that it's compatible with analysis. We do some form of batch alignment, clustering and dimensionality reduction, and then the kind of quantitative statistical analysis. And we'll go through all of these in just a few minutes with a few examples from our studies. But there's a couple of important features about the package, which I think highlight why this is potentially useful. The first is that it is, as far as it goes, reasonably easy to use, and it's kind of end-to-end. So we don't at the moment do compensation in Spectre. That might change soon, but at the moment we just assume that it's been compensated on the machine or in FlowJo if that's what people prefer. You can export the population you care about, and then everything downstream of that we can handle within Spectre. It also uses this package in R called data table, which is explicitly designed to deal with large data sets and to process them very efficiently. And this for us has been very important in terms of the way we implement this particular package. The data structures that we use within Spectre are extremely simple. A lot of packages out there that deal with single cell data or cytometry data of whatever kind, they enforce a certain level of structure so that you can organize things in certain ways and keep certain metadata. But what we've found in a lot of our work is that all of this additional structure creates limitations on the way that you can interact with these data sets. And fundamentally, what we ended up doing is basically just creating a giant table of cells versus features, which means that we can interact with any kind of data from any kind of platform, which is really quite handy. And we've incorporated some batch alignment strategies within Spectre itself. At the moment, there are a number of batch alignment tools, but they're all kind of, I suppose, existing on their own. And so you have to kind of go from one package to another, figure out what kind of table and what kind of data requirements you need to be able to run it in that, then take it into another program. So what we've done is nothing special. We haven't invented any new batch alignment tools. We've simply gone and found the ones that we think are the best, or at least the best in our experience, and incorporated them into Spectre so that it can utilize the very clever tools that people have come up with in a streamlined fashion. And that's sort of the big picture idea of what we're trying to do here. If you want any details on this, we'll go through this in a minute, but the preprint and the website where our code is should have all this information as well. All right, so this is just a point on how the data is structured. Keeping cells and whatever kind of analysis we do in just a giant table for us is actually really helpful. One of the common things that I've heard colleagues of mine say is that when they're trying to work with complex objects, if say they're a flow person and they're used to dealing with FCS files or flow frames, and then they're trying to deal with some single cell data, which is in a Surat object format or a single cell experiment object, they have to sort of find some way of converting one data type into another. And it is entirely doable, but it's reasonably irritating to do so. if you're not familiar with it. Or if you just kind of want to have your single cell data in a table, so you can use some simple plotting functions from R, save it as a simple table, things like this. Now these are all achievable goals, but in terms of running analysis and trying to do different things, it's really quite handy for us to be able to just say, okay, all we're going to do in terms of structure is have a giant table. Each row is a cell, and each column is either a feature of the cells that we're measuring, so like one of the markers from a flow experiment. It might be an entry for which sample this is, which group, which batch, etc. Then any kind of analysis, so which cluster it belongs to, what the t-SNE parameters are, etc. The other nice thing about data table is that the processing and reading and writing speeds are really very good. One of the reasons for this is data table utilizes some low-level parallelism. And so it can, depending on how many CPUs your computer has, it can basically speed up whatever process it's doing by dividing it into kind of chunks that run simultaneously. So if, for instance, if we're reading files into R, if we're using a single thread, for instance, so using only one core of a CPU, it's, for whatever data set this takes 10 seconds. If we're using 8, it's kind of cut it down to, I think, about 3 seconds. Now, 10 versus 3 seconds is not a huge difference, but this scales linearly. So if this was 10 minutes, it would take 3 minutes using 8 threads. It doesn't seem like a huge issue, but when you're trying to analyze all kinds of different experiments and sort of quickly iterate between different collaborations, those minutes end up, amounting to quite a lot of time. So being able to increase the speed of things like reading and writing files is, I think, actually hugely advantageous, as well as operations like filtering or, organizing data in some way. And this is kind of the big selling point for data table in terms of what they do. So it just speeds up the little things, which, it doesn't amount to like most of the time that we would spend working with the data, but it does make a difference, I think, in the end, being able to just do a lot of these things quickly and simply, which is nice. Now, most of this analysis utilizes clustering and dimensionality reduction. And just in terms of a very brief introduction for anyone who's not familiar, clustering is quite a simple concept. Cells that are similar get collected into a group and given a label. So cells that are, let's say, consistent with a T cell phenotype get grouped together and given a label cluster 1. Cells that are similar to a B cell phenotype get grouped together, given a label cluster 2. And we do this across an entire data set. And then this can be plotted in some form of dimensionality reduction, like what's on the left here. So this is a t-SNE plot. The essential way to think about what we're seeing here is each dot here represents a cell, and the cells are arranged in 2D that sort of represents the higher dimensional relationships. And so it's not exactly as I'm describing it, but the cells that are close together in these kind of islands, we infer them to be similar in phenotype. Cells that are further away tend to be less related. Now, that's not absolutely true in t-SNE. The global positioning of things doesn't necessarily tell you how similar two cells are, but the kind of local relationships on this t-SNE plot are meaningful. So what we can do is look at a plot like this. We can see, in this case, the cells are colored by their cluster, and we can go and look at marker expression patterns on this data and figure out what the cells are. So in this case, this big cluster down here, 38, it has high levels of expression of Ly6G, so I would infer that these are neutrophils. Now, one thing that we have to do in this space is be a bit strategic about how we utilize these tools. From a number of papers around the place, including one of our own, this first one here was a great example by Lucas Weber, where you can look at essentially how accurate an algorithm is versus how long it takes to run. These are both important things. You could make an argument that, well, accuracy is the thing that matters in the end, but actually being able to run things quickly so that you can see the result, assess it, decide if you need to make a change, run it again, run a few versions and see how they compare, is actually really important to the way that we do this kind of a work. So runtime, or how quickly an algorithm can do its job, is actually really important. The result of this study was there's a couple of standout options, particularly FlowSOM down the bottom here, which has a high accuracy and a low runtime. So it operates fast and it's very good, essentially. So most of our work, we use FlowSOM, but we do change to other algorithms depending on exactly what we're trying to achieve at the time. But this also applies to dimensionality reduction. So when we run things like, t-SNE or UMAP, various popular versions of dimensionality reduction, these have a similar kind of paradigm. Some work better than others, some are faster than others, but there are some limitations with dimensionality reduction that are a little bit different. As we increase the number of cells being added onto a plot, we end up in this situation, which we refer to as data crowding. So if you look at the bottom example here, if we're only running something like 10,000 cells, this isn't too bad. As we start increasing it and getting closer to 1 million, the data becomes like a big ball. There are various ways to address this and variations of t-SNE that allow it to optimize this process slightly better. But what happens here is we hit kind of a point of it's not a scalable process necessarily, whereas clustering often is. And so we need to think a little bit strategically about how we utilize these tools in this context. So we'll come back to that in a second. The other big challenge that we have to try and address is dealing with batch effects. Now, batch effects, essentially, we can visualize in a plot like this. So this is a UMAP plot, similar to a t-SNE plot, and we've got cells from 2 different batches, so batch A in red and batch B in blue, or green, whatever that is. Now, what ends up happening here is the cells in batch B have maybe slightly higher levels of a few markers, maybe slightly lower levels of a few other markers, because the antibody stain, the antibody master mix was made differently, and pipetting error means that there's slightly different amounts of antibody. Maybe it was slightly warmer that day, whatever. The point is that the actual signal on those cells is slightly different compared to batch A. So what that means is that on a plot like a UMAP plot or a t-SNE plot, cells that are nominally, in this case, neutrophils up the top, will split, and they will have batches sitting in different places. Now, if we cluster this data, what we might end up with is, neutrophils from batch 1 in, let's say, from batch A in cluster 1, neutrophils from batch B in cluster 2. And so what we end up doing is splitting them into separate populations. And if we quantify that, then what we're actually doing is measuring difference between batches, not experimental groups. And this is something that we don't want to do. At the extreme end of this is if we have enormous batch differences, it isn't just a subtle shift in where these cells sit, it's actually an entire reorganization of what the structure of the data looks like. So trying to analyze the top plot, you could argue like, well, it's not too bad. You can sort of the cells group together and that's okay. The bottom plot would be a nightmare, particularly if you had more than 2 batches and you were trying to make sense of this kind of a mess. So there are a few different approaches for doing this. The field of single-cell seq has got a whole range of them, and I think these have been driven largely by necessity because this has just purely been absolutely needed in this field. In cytometry, there's been quite a lot of development in this space as well, but I think not the same sort of range of options out there. But there are some options out there which we think are really quite good and in our testing have stood out from among the others. In particular, cytonorm down the bottom here by Sophie van Garsen, who also wrote the FlowSum algorithm. Cytonorm has kind of come out as our favorite way to do this, but there's a few other options which we've incorporated into our approach as well. Just to give a brief idea of how this works, and cytonorm in particular, we can take, let's say, a concept where we've got a study, we've got some healthy volunteers and some diseased individuals, let's say it's COVID, and then we can also have a reference control individual. And normally we'd take, let's say, a single vial of blood from each individual in this study. The reference control donor will provide multiple vials of blood. So let's say 3 vials, and these are all biologically identical. They were all taken from the same individual. As we process different batches of samples, this just depends on, I guess, when patient samples are available and when we stain them. Every time we do a batch of samples, we want some healthies and some disease, and we'll take one of the reference samples in that batch as well. So each batch has some healthy and some disease in them and one of each of the reference controls. Now these reference controls, because they're biologically identical, the differences that happen in each of these batches, any differences that we can see, are derived from the assay. So either the way that they're stained or differences on the instrument when they're required. And so what this allows us to do is model the batch effects that are occurring, because the only way that they can be different is things that we've changed in the assay or in the way the samples have been acquired. So if we take just the reference controls on their own, we can essentially cluster them, figure out the cells that are consistent with each other and group them into these meta clusters. We can then figure out the quantile conversions so that we can adjust the data so that they end up in the same feature space. So we'd call this alignment. We've basically shifted the signal so that they line up with each other. This is using a quantile distribution system. So the assumption here is that each reference control has the same number of neutrophils, the same number of T cells, etc. And it uses that assumption to shift the data so that you can actually align them and have the batches sort of intermix with each other. When we've then got the rest of the samples, these can be mapped or kind of classified into the meta clusters we had from our reference samples. Once they've been assigned to the meta clusters, we can apply the same rules that we learnt from the training above, and then we can align the rest of our data set, resulting in mixed batches. And then if we see any differences in the data set, we can look at whether these are due to their experimental groups. So what we've essentially done is remove technical variation to the extent that we can from the experiment, which is really quite important. So this is what it looks like. If we've got small batch effects, we can align the data reasonably well, and that all intermixes. If we have very significant batch effects, we may do this a little differently. We may do this through a couple of steps, but ultimately we can get the data to intermix. So the batches have become integrated. Any differences that we see between samples should be due to the experimental group, not the batch. Now, importantly, incorporated not just cytonorm, but a few different approaches that have been published over the last few years. So rather than having to sort of use a different program for different approaches, it's all a one-stop shop, essentially. So you can arrive, test out different batch alignment options, figure out what makes the most sense for the data set, and then get on with the rest of the analysis, which we like. So just an example of what this looks like in the real world. Here we're looking at viral encephalitis. This is kind of work that stemmed from my PhD. So if we take, let's say, brain cells that we've isolated from mice. So let's say in this example, there's 6 mock-infected mice, so otherwise normal mice, and 6 mice that have been infected with West Nile virus. And we've taken these brain samples 7 days after infection, where there's a large immune infiltrate into the brain. So we take our large data set, we do some clustering with FlowSum, which is sort of our preferred algorithm. And because the FlowSum algorithm will tolerate very large data sets of millions and millions of cells and operate on this very quickly, but the dimensionality reduction steps might struggle with some larger data sets, what we can do here is we take the data after we've clustered it, we could subsample it, so take representative cells out of that data set, and plot that on UMAP, on a UMAP plot, a dimensionality reduction plot. What this means is that we can investigate what's on the plot. So in this case, in sort of C here, these cells are colored by either the cluster or the population we've labeled it as, but this represents the cell. So this might only be, let's say, 10,000 cells, but this represents the potentially millions of cells that were in the that have been clustered. And so what we know is that if on this plot, cluster 1 is, let's say, resident microglia, cluster 1 in the large data set also represents microglia. And so even if we have small numbers of cells, like in this case, this T cell island in red is quite a low number of cells. Maybe it's, I don't know, let's say it's 50 cells. We want to have large numbers for robust statistics. And what we can do here is essentially say, okay, well, if we know, let's say cluster 4 is our CD4 T cells, and there's only 50 of them on the plot, cluster 4 in the large data set is going to be the same population, but in this case, we might have thousands to millions of cells, and we can get more robust statistics out of the data that way. So we can use this sort of subsample data for plotting, figure out what all our populations are. Once we've got that, we can then go and do our quantitative work with reasonable statistical power. In this case, what we find is not particularly it's basically, 7 days after infection, you get a huge infiltrate of immune cells, kind of everything goes up. The most prominent infiltrating cell into the brain in this case is these sort of monocyte-derived macrophages, which come and kind of mess the brain up as they try to fight off the virus. But this is kind of a good example in terms of how we can do this analysis. The summary data functions that we use in Spectre are all based on data tables aggregate functions, and it very, very rapidly allows us to summarize number of cells per sample, the frequency of the sample those cells comprise, the MFI of different markers, how many cells are expressing different markers, and sort of create these sort of summary plots, either individual bar graphs or violin plots like this, or things like kind of global heat maps or PCA plots. But we can do all of that very rapidly and automatically, which gives us not only a lot of power, but a lot of speed in terms of how we can do this analysis fairly seamlessly. We can also then take advantage of that approach for some more complex situations. So part of my work was looking at the bone marrow, and if I plot an entire bone marrow sample using something like UMAP, I can see kind of all my different lineages branching out in different areas here. But these little cells in red are progenitor cells. Now on this plot, there's not a whole lot of them. There's, you know, I don't know, maybe there's a couple of hundred on there. I would actually like to extract the progenitor and stem cells themselves, isolate them, and, you know, analyze them separately. So what I can do is figure out which clusters represent the stem cells. I can extract them not from this plot, but actually take them from the larger clustered data, figure out, you know, cluster 10 and 12 represent the stem and progenitor cells. I can extract them and do a whole new analysis now with thousands of cells and work out, you know, what lineages I think are occurring within just this small part of the whole data set. So we can do kind of this analysis at multiple levels, which allows us to have quite a bit of granularity for the way that we do this work. And this is important because, I mean, subtle differences, particularly with progenitor work in this case, you know, subtle differences matter. And they matter in terms of the way that we're able to split up lineages, say, go on to sort cells and figure out what they are. These subtle differences are important, but they're not obvious when you're looking at the data set as a whole. We do need to be able to isolate an element of, say, in this case, the hematopoietic system, drill down on just that element and pull that apart. So it is important being able to do this at multiple levels, and we think the approach that we have here facilitates that to an extent. The other example I have for this is our recent work doing some immune profiling in COVID-19. And this was done with a group from the Doherty Institute in Melbourne. And Melbourne is where we had our sort of largest outbreak of COVID over a couple of waves. So this was a profiling study from the first wave that Melbourne had. We were fortunate to not have quite the same outbreak in Sydney. We didn't do super well, but we definitely got lucky, I think, in terms of not having the same kinds of numbers. So the group, the Doherty, were able to access quite a decent cohort of COVID patients. But what this ends up generating is, you know, quite a large amount of data. So in this case, we did 3 separate panels on, this is just on a conventional flow cytometer, and 3 separate panels based on what they had running at the time. So each of these panels had somewhere in the order of 80 to 100 million cells total from all the patients. So we did the same process as before. We do some batch alignment to deal with some of the batch effects that came up. We do the clustering on this data set, which is really quite large, but FlowSum tackles it just fine. We can then sample this, do our UMAP plots, figure out what populations we have, annotate the clusters, and then sort of take the data out of that clustered set so that we can do our quantitative analysis with reasonable statistical power. The end result, without going into kind of too much detail, is essentially what we found is that in acutely infected patients compared to healthy or convalescent patients, I mean, surprising no one now that we know more about COVID, but essentially what we're seeing is, you know, high cytokine production as well as kind of activated immune cells, which is not especially surprising in a viral infection. But we have got to really, I suppose, get some granularity on the time dynamics of this kind of response over such a large cohort, and then see what this is like in convalescent patients, kind of, you know, up to about 100 days after their initial symptom onset, which is really quite cool. We're also able to do some more comprehensive profiling in the ICU patients, so patients with severe disease compared to patients who were being treated in the ward or treated as outpatients. And for the most part, consistent with many other studies, is that we do find a hyperactivation of many immune subsets in this case. So excessive kind of production of cytokine and excessive activation of a number of these cell types, as well as the appearance of a number of unusual cell types, such as immature neutrophils in the blood, which is kind of one of the hallmarks of this disease, in particular as it progresses towards ARDS and sort of tissue damage in the lung. So this was quite good in terms of, you know, I suppose demonstrating what we can do. Some of this was not necessarily just, you know, doing clustering. but it's kind of the ability to take either data we've clustered or data that we've gated and rapidly generate a lot of these summary statistics all in a single workflow, which gets us to this answer very quickly. Because these are done in different panels as well, not only can we analyze them separately, but this data can then be brought together for our sort of more global analysis, which paints us a bigger picture when we're trying to look at, the same patients that have got different measurements and integrate that data some way to give us some meaningful conclusions, which is really quite helpful. All right, so the last thing I want to talk about is some areas of development for our package and where we're trying to go from here. And there's two that I want to talk about and just give a little bit of an overview on. One is in spatial analysis, so for imaging data, as well as some sort of future thinking about data integration. So spatial analysis to begin with. At our facility, we have an IMC, or an imaging mass cytometer. This is the Hyperion system from Fluidigm. This is quite fun. Essentially, it's a cytoff with an imaging module on the front. You use a laser ablation system to blast off little bits of tissue from a slide, which has been labeled with metal conjugated antibodies. Whatever material is in that spot basically gets sucked into the cytoff and analyzed as per usual. But rather than going cell by cell, as we normally do with the cytoff, we're going sort of 1 micron spot by 1 micron spot. And we can basically do that across an entire tissue section, taking in all that material into the cytoff. And it generates some pretty cool imaging data, up to about 30 to 40 plex currently. And most of our panels are somewhere in the 25 to 30 range at the moment. So this is quite fun. We can get some very pretty pictures, but what we do want to move towards is getting something quantitative out of this. Now, this can be a little bit tricky. There are a number of papers that have been published on this recently, and HistoCat was kind of the premier tool that was released for use with IMC data. Fundamentally, what this looks like is you need to do some kind of cell segmentation. So essentially on the image, there needs to be a border drawn around each cell so that it can say, okay, all the information in this area belongs to cell 1, and it can then make a data point out of that. Once you've got a data point per cell, you can do things like clustering, t-SNE, gating, or what have you, and then you can perform some kind of spatial analysis. So ask questions about how the phenotypes that are present in the data relate to the regions in the tissue or the spatial relationships therein. Now, what we did as a result of our work with Spectre is we made some modifications and created this is sort of in a beta test phase, but Spectre map, which is like an extension of Spectre explicitly to work with spatial data. And we've been working with that for the last few months, and it's been steadily improving. The first thing that we do as part of this workflow is segmentation. So we use Elastic for this, which is an open source software tool. What we can do is take, in this case, DNA. So we can take signal from DNA or various cytoplasmic markers. We can draw, and like physically draw, kind of like with Microsoft Paint, areas that represent background or cell borders or the nucleus. And when we allow the computer to look at that data, it can use that as training, and then it can predict what it thinks each pixel in the image belongs to. So in this case, it's predicted, I'd say fairly accurately, that these nuclei are all nuclear signals. It's worked out where the cell borders are, and it's figured out that this kind of space in the middle is all background. So once we've trained the classifier sufficiently to be able to work out where the cell boundaries are, it can give us some boundary probabilities and actually split the image up into individual cell segments. Once this is done, we end up with essentially data points. And so now everything within the border of each cell can be averaged or integrated, and this then gives us a table that we can analyze. So this is just what some of that data looks like. This is an IMC image taken from some mouse brain that's been infected with West Nile virus. So this is the hippocampus that we're looking at. And in this case, each dot on here is kind of, in the center of a segmented cell, and the little yellow outlines are the actual border of that cell. So this is neurons through the hippocampus of the brain, and this middle image is NS1 expression on those cells, which is the viral protein produced by West Nile virus, which is quite a pretty image. And then on the right here is CD11b. So this is expressed both on microglia in the brain, but also infiltrating cells that come in and cause a whole lot of damage. What we can do is a couple of things. One thing that we have to keep in mind is that when we do the segmentation process, there is a whole lot of cellular overlap that we need to try and deal with in some way. What this means is that if we look at normal flow data, we'd sort of think, okay, well, these are CD20 expressing, so these are B cells and these are T cells down here. But these double positive CD20, CD3 cells are a bit of a mystery. In this case, if you have, let's say, a T cell that's surrounded by B cells, that T cell will gain a whole lot of signal from the B cell surrounding it, and it will express, in this case, just as much CD3 as CD20. Now, this kind of puts you in a difficult situation because it's like, well, you don't actually know just from the data points which is which. You can't say, well, this is a T cell in the B cell zone, because when you average the data that's being expressed within that cell, it doesn't really help, as you can sort of see here. So we do have some slightly different ways of doing this, because spatially we can determine which cells we think are B cells and which cells we think are T cells. And so we actually can change the system a little bit and pre-classify cell types based on the spatial data, not just the actual data points themselves. This can be a bit of a tricky process, but it works quite well for us. And we can also do this for regions of the tissue as well. So we can pre-classify areas of the tissue, which helps us with some of our spatial work. What we can then do once we have all this information is we can take our individual sort of channel data from these images, our region masks or our cell type masks, as well as our actual cell boundary masks. We can integrate all that data together and sort of go through our normal analytical process, so to speak, generating our clusters on our cell types. And once we have that output at the end, we can start asking some spatial questions, looking at how we can identify how different cell types relate to each other in a spatial context. And does that change, say, between a healthy sample and a sample from a disease patient, which is really quite fun. The last area of development for us is looking at data integration in the context of things like the Human Cell Atlas. This has been a sort of a background project for the last couple of years, as we've tried to see for a couple of years how things like cytometry can both contribute to and benefit from the Human Cell Atlas. And there are a couple of slides which I've borrowed from Rahul Satija, who kindly gave me permission to use them. But a nice way to think of how this works and what the Human Cell Atlas project is similar to what was true for the Human Genome Project. Essentially, prior to this, there is no map of the genome that we can use to understand any of the genomic or genetic work that we're doing. The result of the Human Genome Project is a reference map. So then when we're dealing with genomic data of lower quality or lower reads, we can map it onto a known reference. So this is a fundamental, change in the way that genetics and genomics is able to be done, by having some kind of assembled reference map that you can then refer back to. This is actually the same as what the objectives of the Human Cell Atlas project are, is to create essentially a reference map at single cell resolution of all the molecular features that can possibly be measured. So that way, when we look at new data sets, we can use a reference map to figure out what's going on. And one of the most recent examples from Rahul's lab is doing this classifying populations in samples from COVID patients. And so if you have a reference map that not only includes healthy patients, but also a variety of disease situations, you can classify cells in a way which is not only biologically relevant in the normal situation, but is also relevant in a kind of a disease situation as well. And so what this attempts to do is essentially remove the need for us to go and almost manually go and decide which cells are which. We can have a data set, we can integrate it with one of these atlas data sets and have it pre-classify cells into populations based on what it thinks its closest match is, which is really quite powerful. Key to this is some kind of integration strategy which allows us to work across data sets. Work across technologies. And just to make an example of this here very briefly is we took some spleen cells from a mouse, did a panel for our LSR platform, LSRX platform, which is kind of like a symphony, our Aurora and our CyTOF are using separate panels. And so if we try to analyze this data together, as you'd imagine, this is kind of like batch effects. There's huge technical differences between each of these technologies. And so the data that comes flow or mass or spectral is, in all kinds of different places. But using our alignment approaches, particularly cytonormal on the end here, we can actually get the flow and the spectral and the CyTOF data to integrate in a way that's reasonably effective. So this is quite good. We can get data from different technologies into a single feature space, which allows us to analyze them together. What this means is that if we have, for instance, two data sets, in this case, one is labeled with cell types and the other isn't. So let's say we've got a reference data set which is pre-labeled and a new data set, let's say from a COVID patient who's just come in. What we can do is use a label transfer system with a classifier, which we'll talk about in just a second, and we can fill in the blanks essentially. So now the gray cells here from dataset B, which aren't labeled, have become labeled based on, in this case, their nearest neighbor. The way this works in a little bit more detail is if we take dataset A and dataset B and we sort of split them into these two panes. Dataset A has labeled cells, this is our reference, and dataset B is our sort of new dataset. If we use a classification system, in this case, we're just using a k-nearest neighbor classification system, which is really quite simple and is not as elegant as some of the single cell approaches being done at the moment, particularly with tools like Surat. But we've found that this system actually works pretty well, probably because of its simplicity. Essentially, the cell in this data set will be in one of the data sets will be compared and matched to its nearest neighbor, essentially. So if you have a CD4 T cell, it will go and look for which cell in the other data set is the most similar, and it will pair it with that. Once it's been paired, you can essentially transfer the label. So if your query cell has been matched to a CD4 cell in the labeled data set, then it will gain that label. And what that results in is two labeled data sets. So the reference data set, which is the same as was before, and our new data set, which now has gained the cellular labels based on a classification system. This data can either then be merged back together, if that's the point of the experiment, or the new population can just be analyzed in and of itself, depending on what the requirements of the experiment are. So this is kind of the future for us, is going from, just clustering and, cool ways of plotting and sort of being able to annotate cells to having a system where we can take new data, integrate it with some reference atlas data set, and then give a machine learning sort of approach within Spectre in this case, so that we can automatically label cells with some kind of predictive score along with it, so that you can look at the data, say, cool, this has worked out well, or this prediction is rubbish and I'd rather do it on my own. We think this is a flexible and future thinking way of approaching this kind of work, which we're pretty excited about. Okay, so that's all I've got in terms of our applications. I would just particularly like to thank Felix and Giovanna, who are my Felix here and Giovanna here, who are the co-developers of Spectre, as well as the labs that we've worked with and applied this to in a number of contexts, and our funding bodies as well. If you have any, if you want to go and find out some more information, there's sort of our links here on the bottom right as well. So thank you very much for your time. I'll happily take any questions.
All right.